21st PSI online lecture series. It's an effort to support the undergraduate students who may need some supplementary support in terms of physiology teaching. This is an effort by the Education Committee of Physiological Society of India, and on behalf of Physiological Society of India, I welcome you all for this session. Today we have guest speaker, Professor Abhinav Dixit. Professor Abhinav Dixit is currently working as Dean Examination Professor and Head of Physiology, AIIMS Jodhpur. He is a product of prestigious medical college, Maulana Azad Medical College and University College of Medical Sciences, Delhi, commonly known as MAMC and UCMS, Delhi. He also obtained his DNB Physiology and MBA in Healthcare Management. He is a fellow of Royal College of Physicians, Edinburgh, Royal Society of Biology, UK, and International Medical Sciences Academy, and member of National Academy of Medical Sciences, India. He has established and is in charge of Cognitive Neurophysiology Lab at AIIMS Jodhpur and has funding and collaborative projects with various institutions. He is also an innovator with patents. He is the winner of various awards for innovation, including Indo-Sweden Healthcare Innovation Challenges 2020 and 2022. He has also developed products with industry. Dr. Dixit is currently editor Physiology of Indian Journal of Physiology and Pharmacology, IJPP commonly known, and society representative of the Physiological Society, UK. He has publications in various international and national journals and is subject expert at various universities and recruitment bodies. Because of some technical issues, we are not able to take Professor Dixit live online to this session. However, he is kind enough to share his lecture with us, and we will be playing that lecture on his behalf. And I know with the IPL starting at the same time at Calcutta, we will be hardly any audience today. That's unfortunate. Anyway, it's good. I don't know actually how to, but it is. We will be welcoming all the questions. Those who are uh, offline or those who will be enjoying this stream afterwards, they can send queries to Professor Dixit. He has given his consent, and so without much delay, I will be playing his recording, sharing this recording, and hope you will enjoy.
about the physiology of cerebellum and its role in motor function, it is important to know the neuroanatomy of cerebellum. Cerebellum is what our school books have been calling as the little brain, and all of us have read the word little brain in our school lives. Cerebellum is located in the posterior cranial fossa behind the fourth ventricle, the pons, and the medulla oblongata, and it is separated from the cerebrum by a fold of dura mater that we call as tentorium cerebelli. Anatomically, cerebellum is divided into three lobes by two transverse fissures. The first being what is known as the primary fissure that separates the anterior and the posterior lobes. The anterior and the posterior lobes together form what is known as the body of the cerebellum. The posterolateral fissure separates the cerebellum from the small flocculonodular lobe. The cerebellum is relatively small in size, and if you look at it in comparison to the cerebral cortex, it weighs only as much as 10% of the cerebral cortex. But if we look at the surface area of the cerebellum, it is about 75% of the cerebral cortex. Now, this shows that functionally and anatomically, the cerebellum is an important part. Therefore, in spite of grossly looking to be small, because of the folds that is as, its surface area is as much as 75% of the cerebral cortex. The fissures divide the surface of the cerebellum into narrow leaf-like bands that we call as folia, and each folium is made up of a central core of white matter surrounded by gray matter. And the arrangement of white matter is such that it appears like a branching tree, and hence it is also known as arbor vitae. These two diagrams basically show what I've been talking about in my previous slides. The cerebellum is located behind the pons and the medulla and the fourth ventricle, divided into an anterior and a posterior lobe and a flocculonodular lobe. Also, as you can see in this diagram, the classic branching pattern or the arbor vitae is clearly visible in this diagram. It is important to understand that the cerebellum is connected to the rest of the brain or the central nervous system through certain pairs of structures that we call as the peduncles. And since there are three pairs of these peduncles arranged from up to down, we name them as a superior cerebellar peduncle, a middle cerebellar peduncle, and an inferior cerebellar peduncle. Now, each of these cerebellar peduncles has tracts which are running to and fro from the cerebellum and helping in transmission of signals and connection of cerebellum to the other parts of the brain. The superior cerebellar peduncle connects the cerebellum to the midbrain and the thalamus. The middle one is connecting it to the pons and has only the contralateral fibers coming from the contralateral pontine nuclei, whereas the inferior cerebellar peduncle is the one that is connecting the cerebellum to the medulla oblongata. And it is this inferior cerebellar peduncle that is transmitting the afferent fibers from the brainstem and the spinal cord, as well as the efferent fibers to the vestibular nuclei from the cerebellum. Like all parts of these areas, the cerebellum also is made up of a gray matter and a white matter. And we, in case of cerebellum, divide into a cerebral cortex and the deep nuclei. The deep nuclei go by the name of dentate, globose, emboliform, and vestigial. And as we go further into this lecture, I'll be talking about the relationship of the cerebral cortex as well as of the deep nuclei. Now, this diagram is basically showing us the relative locations of the cortex and the deep nuclei. And as you can see that the deep nuclei are deeply seated here, whereas this is the cortical surface of the cerebellum. If we look at the cerebellar cortex, there are different type of cells that are present in the cerebellar cortex, and these cells are arranged into different layers which are functionally specialized. Now, if we have to have a view of the cerebral cortex from out to in, the three layers that we can see are what are known as the molecular layer, the Purkinje layer, and the granular layer. And each of these layers is made up of a particular type or more than one type of cells. For example, the molecular layer consists of what are known as the stellate cells and the basket cells. The Purkinje layer is made up of the Purkinje cells, and the granular layer is made up of the Golgi cells and the granule cells. Now, when we talk of the cerebellar cortex, we need to understand that the cerebellum is functioning as a comparator. Basically, it is comparing the original motor plan that has been made by the motor cortex and the real-time input that it is receiving from the proprioceptors. So the main excitatory input that comes to the cerebellum comes from two types of fibers, which are known as the mossy fibers, which are originating from the spinal cord and brainstem and are ending in the granule and Golgi cells at the granular layer, and the climbing fibers that are originating from the inferior olivary nucleus, and they are basically synapsing with the Purkinje cells in the molecular layer. Now, as I said previously, that the granular layer is made up of granule cells and Golgi cells. And these are the cells that are receiving the primary input from the mossy fibers. Also, we need to understand that when we talk of cerebellum, there is an entity that we call as the cerebellar glomerulus, which is nothing but a synaptic complex which is made by the mossy fiber, the axon terminal of the mossy fiber, and the dendrites of the granule cells and the Golgi cells. Now, important thing to understand is that, which I'll be showing in the subsequent diagrams and the figures, is that the axon of the granule cells extends into the molecular layer, and there it bifurcates into a T-shaped division, wherein the fibers of the T's run parallel to the long axis of the folia, and hence they are also known as the parallel fibers. And it is these parallel fibers that are forming the excitatory synapses on the dendrites of the Purkinje cells. So we need to understand that the input is coming from through the mossy fibers to the granule cells. These granule cells are further dividing and forming what are known as their axon is dividing into T-shaped structure and the parallel fibers. And it is these parallel fibers that are going further and exciting to the Purkinje cells and the basket cell. Now, the Purkinje cell layer is comprised of the Purkinje cells, and the Purkinje cells are the largest cells in the central nervous system. Now, it is important for us to understand that the output of the cerebellar cortex is through the Purkinje cell layer. Now, I'm not saying the output of the cerebellum, I'm saying the output of the cerebellar cortex. Now, this output of the cerebral cortex is through Purkinje cells, which are basically receiving the excitatory input from the climbing fibers, which I talked of in the previous slides, and from the granule cells through what is the parallel fibers. Also, the Purkinje cells receive inhibitory input from the stellate cells and the basket cells. The important thing to understand is that as I said earlier, also I'm repeating it again, that the output of the cerebellar cortex is through Purkinje cells, and they give their output to the deep cerebellar nuclei or to vestibular nuclei, and it is this output from the Purkinje cells which would further modify the net output of the cerebellum that is coming from the deep nuclei. Now, if you look at the molecular layer, the molecular basically is consisting of basket cells and stellate cells, and these are cells which are inhibitory to the Purkinje cells. But the thing to remember here is that these cells are also getting their input or excitatory inputs from the parallel fibers. So the parallel fibers are basically supplying the basket cells as well as the Purkinje cells. The Golgi cells also project into the molecular layer, and again they are also receiving their excitatory input from the parallel fibers. This diagram that I have taken from Kendall is basically showing us the structure of the cerebellar glomerulus and the arrangement of the various layers and the arrangement of the cells in the various layers of the cerebellum. Now, from the functional point of view, it is also important for us to know the neurotransmitters that are involved in the transmission in the case of cerebellum. And as I said, granule cells were the ones which were giving out their fibers and forming the parallel fibers, and the parallel fibers were going and exciting the other cells, whether we talk of the basket cells or whether we talk of the Purkinje cells. So the key thing to remember here that the granule cells are excitatory in nature, and the neurotransmitter that they are using or they are secreting is basically glutamate. Whereas the other cells, we talk in terms of Purkinje, Golgi, basket or stellate cells, all of these cells are releasing GABA, and hence all of these cells are inhibitory in nature. Now, if we look at the various cells and the fibers that I had talked of in the previous few slides, we come to look at the cerebellar circuit that is playing a role in or that is present in the cerebellar cortex. Now, this cerebellar circuit, if we talk of, is basically the cortex is receiving the inputs from two types of fibers, which are the mossy fibers and the climbing fibers, out of which the climbing fibers are exciting the Purkinje cells directly, whereas the mossy fibers are stimulating the granule cells, which are further giving rise to the parallel fibers, and it is these parallel fibers that are going and in turn stimulating the Purkinje fibers. Now, if we look at the other two types of cells that are present, that is the basket cells and the stellate cells, we see that they are also excited by the granule cells via parallel fibers, and it is these basket cells that are going and in turn inhibiting the Purkinje cells. So this kind of inhibition is what is known as feed forward inhibition. Now, something to understand here is that the function of the granule cells is being basically regulated by the Golgi cells. Now, these Golgi cells are being excited by the mossy fibers as well as the parallel fibers, and they are inhibiting the transmission from the mossy fibers to the granule cells. So they're in a way regulating the function of the granule cells. Now, as I said earlier, that the final output of the cerebellar cortex is through Purkinje cells, and this output is going to the deep nuclei. And this output of the Purkinje cells to deep nuclei is inhibitory in nature. And it is from these deep nuclei that the final output of the cerebellum is going to the brainstem and the thalamus. But it is important to understand that the final output is excitatory. So the output from the cortex to the deep nuclei is inhibitory. The output from the deep nuclei to the brainstem or thalamus is excitatory. And how is this happening? This is happening because of the sum total of the various inputs that are acting in the at the level of the Purkinje cells to regulate their output. Now, I have been talking a lot about cells and their connections with one another. This is a figure that again I've taken from Genome, which is actually showing all that I have been speaking of in the last few slides. Now, if you look at this figure, we see that the mossy fibers are coming, and they are obviously sending a branch to the deep nuclei, which is excitatory to the deep nuclei. Also, the mossy fibers are causing excitation of the granule cells as well as the Golgi cells. Now, it is these granule cells that are moving up, the axons are moving up and dividing into and forming the T-shaped structure that I was talking of in the previous slides. This is that T-shaped structure. And these then form what are known as the parallel fibers. Now, it is these parallel fibers that are going and that are stimulating the Purkinje cells directly. And as you can see in this figure, they are also stimulating the basket cells. Now, these basket cells in turn are going and inhibiting the Purkinje cells. So this kind of a system wherein the granule cells are in turn stimulating the Purkinje and stimulating the basket, and the basket are further going and inhibiting the Purkinje cells itself, is what is being referred to as the feed forward inhibition. Now, we can also see that this diagram shows us the climbing fibers. These climbing fibers also go and stimulate the Purkinje cells, and they also stimulate the deep nuclei. As is evident from this figure, the output of the cortex to the deep nuclei is solely through these Purkinje cells, and these Purkinje cells are the ones that are going and inhibiting the deep nuclei. Now, the net output of the Purkinje cells would be the sum total of the effects of the stimulatory and the inhibitory synapses that are acting on these Purkinje cells, and the net output, which is excitatory from the deep nuclei, would in turn depend upon the net inhibition and the excitation of the deep nuclei through various inputs, whether it is the Purkinje cells, mossy fibers, climbing fibers, or any other input that is going to the deep nuclei. So far, we've talked of the cerebellar circuits. The next thing that we need to understand is the role of the cerebellum in the motor system. And if we look at the cerebellum, it can be divided into three functionally different parts as vestibulocerebellum, spinocerebellum, and cerebrocerebellum. So if we talk of the cerebrum from a functional point of view, and we divide it into the various parts, as I said in the previous slide. So the most primitive part phylogenetically is what we call as this vestibulocerebellum. And it is made up of or composed of the flocculonodular lobe. The word itself has the word vestibulo in it, so it's quite clear from the terminology itself that it is receiving the vestibular inputs and it projects to the vestibular nuclei of the brainstem, and this part is concerned with the balance and maintenance of balance and equilibrium. Now, it also has inputs from the pretectal nuclei and plays a role in eye movements. Now, if we look at the relatively newer parts, we come to what is known as the spinocerebellum, which is made up of the vermis, that is the central part, and the intermediate parts of the cerebral hemispheres, the cerebellar hemispheres. And it is this part that is mainly concerned with the motor movements, and it is responsible for smoothing and coordinating of the motor movements. Now, spinocerebellum receives the input from the spinal cord, and the input that it receives is basically the proprioception and the touch and the pressure inputs. And these inputs reach the cerebellum through the dorsal and ventral spinocerebellar tracts. The vermis or the central part is receiving the inputs from the visual, auditory, vestibular, and other somatic inputs. And the vermis is the part that is projecting to the vestigial nucleus and plays a role in controlling the proximal muscles of the body, basically the trunk musculature and the proximal parts of the limb. And the spinocerebellum is playing a role in controlling of posture, locomotion, and eye movement. Along with this, the intermediate parts of the spinocerebellum receive input from the limbs and go to the interpositus nucleus, and they provide the input to the lateral corticospinal and the rotospinal tracts for the contralateral side of the brain. And this part that is controlling the distal muscles of the limbs and the musculature for the digits. The lateral part of the cerebellar hemisphere is what is known as cerebrocerebellum. And if we look at it from an evolutionary point of view, phylogenetically, it is the most recent part. And it is involved or concerned with planning and programming of motor movements. The cerebellum receives inputs from the association cortices, and it is basically connected to the dentate nucleus and via thalamus projecting to the contralateral motor, premotor, and the prefrontal cortex. The cerebrocerebellum also has a role in cognitive functions, and it is involved in motor planning, such as functions like visuospatial and language processes. Now, if we were to summarize what is actually happening in between the three parts of the cerebellum, that is the vestibulo, spino, and the cerebrocerebellum, we can see that these three parts are projecting to the deeper nuclei, whether the cerebrocerebellum is going to be dentate or spino is going to the interpositus and the vestigial nuclei. The vestibulo is going to the vestibular nucleus, and these are then serving a specific purpose, such as the cerebrocerebellum which is involved in motor planning, is connected further to the premotor cortex. The spinocerebellum that is involved in motor execution is projecting to the motor cortex as well as brainstem, whereas the through the vestibular nuclei, the vestibulocerebellum is projecting to the spinal cord and brainstem and playing a role in maintenance of balance, as well as the vestibuloocular regulation. So this slide basically summarizes the three functional divisions of the cerebellum. As you can see, this is the flocculonodular lobe, that is the vestibulocerebellum, and phylogenetically the oldest, that is concerned with balance and eye movements, and it's connected to the vestibular nuclei. The vermis and the adjoining parts are what is known as the spinocerebellum, and they are involved in motor execution through the medial descending system and the lateral descending system for controlling the movement of the distal parts of the limbs and the digits. And the lateralmost parts is what is known as the cerebrocerebellum, which is involved in motor planning. So if we look at this diagram, we can see the various inputs and the outputs of the cerebellum and the way they are related to each and other. The inputs are basically coming from association cortex, the somatosensory receptors. They're coming from various other parts going to the cortex at the deep nuclei, and the output is then going to the again motor prefrontal motor premotor nuclei, various interneurons, and the outputs are also coming from the vestibular nuclei. Now, if we were to if we were to see this entire picture, we'll see that the cerebellum is doing the function of a comparator in a sense. It is receiving the copy of the motor plans, as well as it is receiving the real-time input from the various proprioceptors, touch and pressure receptors. So, what is cerebellum actually doing if we look at the function of cerebellum? Cerebellum smoothens out the movement, coordinates the movement, as well as it is maintaining the posture and balance of the body. Now, I'll give a simple example that it has happened with all of us, I guess, most of the time. I would say some time or the other, that when we are trying to skip a step while climbing the stairs, it happens that we realize we cannot leave a step and we put our foot down on the step in between. Now, this is one of the simplest ways that I would like to explain the function of the cerebellum. The cerebellum receives a motor plan. According to that motor plan, the body is acting and we decide to skip a step and take alternate steps. Now, while this is happening, the cerebellum is receiving the real-time input from the somatosensory receptors, whether it is the proprioceptors that we are talking of in the joints, and this input is going to the cerebellum. Now, what cerebellum is doing is it is doing a real-time comparison of the motor plan that it has received, as well as the real-time input that is coming from the periphery, and tries to match the two. Now, if it feels that the matching is not proper, it gives out an output which brings about the change in the movement and tries to smoothen out the movement and brings about a proper coordination, which in case of climbing of these steps would be not skipping the step and keeping a foot down, lest we fall from the step if we try to skip it. So this is a summary of the entire physiological function of the cerebellum to smoothen and coordinate out the motor movement. And cerebellum per se is a very important part of the motor system, whereas it is helping in controlling the rate, range, and direction of the movement. Now, this slide is basically telling us the various afferents that are coming to the cerebellum, and as we can see through the various tracts that are coming, the cerebellum is receiving the input from the vestibular apparatus, from the various proprioceptive inputs. It's receiving the input from the muscle spindles, the Golgi tendon organs, from the auditory and the visual pathways, as well as from the other parts of the cerebral cortex, from the pontine nuclei, through the pontine nuclei, and the proprioceptive input from the whole body. And if we just look at these tracts, we again coming to the previous slide, as I said, it's basically getting the motor plan, the real-time input, and it's trying to match the two and bring about the corrective response. If now we were to look at the symptoms of the cerebral disease, the symptoms are nothing but a manifestation of the function gone wrong. So Sorry, excuse me. If we look at a patient of cerebellar disease, the patient would be having cerebellar ataxia. A typical gait would be was similar to a drunken gait. There would be intention tremor. Now, this intention tremor is different from the tremor that we get in Parkinson's disease, wherein in Parkinson's it's a resting tremor that disappears on movement. In this case, it's a tremor that is not present on rest, but it is manifested once the subject tries to do a movement, say trying to grab a cup. This is because again the pathophysiology would remain the same, that the cerebellum is unable to match the two inputs that it is getting, the real time and the plan, and hence the correction of the movement is not proper. So also we need to understand the cerebellar symptoms would involve things like scanned speech, which is disruption of the speech flow and rhythm. Because the cerebellum is now affected and it is not able to do its part properly, the smoothing of the movements is not there. So the patient of suffering from a cerebellar disease would be able to is basically decomposing a movement into components rather than carrying out a movement in a smooth fashion. There is maybe delay in the initiation of the movement because of the fact that the movement is not proper. There is dysmetria in the sense that the patient is unable to perform movements concerned with the proper measure of a distance, because again, since the input are not matching with each other, the judgment is difficult and hence the movement is not appropriate. We also know that cerebellum is involved in regulation of muscle tone because of the fact that it is one of the areas that affects the gamma motor neuron discharge. And a lesion of the cerebellum is therefore a pure lesion of the cerebellum leads to what is known as hypotonia. I'm not going into the details of this circuit because I presume that it is known to everybody, since muscle spindle, gamma motor neuron discharge, and the regulation of gamma motor neuron discharge is known. This slide is basically trying to show us what a dysmetria is. If we ask a patient suffering from a cerebellar disease and we ask him to touch the examiner's finger and then their nose, they find difficulty in doing it. And as this slide shows, that rather a straight pathway, it is somewhat a zigzag pathway because of the fact, again, as I said previously, the function of the cerebellum is not appropriate, the inputs are not matching, and hence the subject finds it difficult to do a smooth movement and take it to a point of where it can be conducted smoothly. Similarly, there is what is we call as a dysdiadokinesia, which is the inability to do repeated alternating movements. A classical example would be alternating supination and pronation, which a normal person would be easily able to do. Whereas in a person with cerebellar disorder, these is difficult for the person to do. So initially we talked of the symptoms. Now coming to the signs, as I said previously, the as the diagram also showed, dysdiadokinesia is something which is inability to perform rapidly alternating opposite movements, and a classical example is the supination and pronation of the hands. There is nystagmus, which is to and fro movements of the eyes, as is understandable since the cerebellum is connected to various other systems, including the vestibular apparatus. Obviously, nystagmus is a feature in cerebellar disease. This rebound phenomena, wherein this is a reflex wherein when the patient attempts to move a limb against resistance and the resistance is suddenly removed, there's a rebound phenomena that we get to see. There is a pendular reflex where after a knee jerk tapping the on the patellar tendon, the leg may oscillate like a pendulum. So as we've discussed, the symptoms are quite easily correlatable to the functionality of the cerebellum and the physiology of the cerebellum. Hence, the patient of cerebellar disease would be presenting with these symptoms and signs as we talked of in the previous slides. Now, the cerebellar involvement can be due to multiple causes. Herein, I'm just trying to highlight, not going into the details of certain cerebellar degenerative diseases that are seen. One being the spinocerebellar ataxia, which is inherited disease due to CAG repeats. Multiple sclerosis is an autonomous autoimmune disease which quite a few patients are being seen now because of the very fact that it's not only a genetic disease, it also has environmental factors, and the antibodies damage the myelin sheath, leading to the injury of the myelin sheath. And hence, the along with the wider picture, obviously the multiple sclerosis just not purely of the cerebellum. It is basically a demyelinating disorder which tends to involve slowly various parts of the CNS, ultimately involving the cerebellum. So cerebellar signs can also be seen in multiple sclerosis. Similarly, in chronic alcohol abuse, between B1 absorption is reduced. Again, this is not a pure cerebellar disease, but yes, a manifestation of a B1 deficiency could be in the form of cerebellar signs and symptoms. So basically, what we tried to summarize in last few slides over the last half an hour is the gross functionality of the cerebellum, the way cerebellum controls the motor movements, and the various cerebellar diseases, the signs and symptoms of various cerebellar diseases, and the physiology behind those signs and symptoms. If I haven't gone into much detail, because the entire lecture was basically to give an overview of the cerebellum and its functionality. The people who are seeing this can refer to various other books which go into much greater depths, which was not possible because of the constraint of time that we have here. But to summarize, I would say that cerebellum is the part of the brain which is responsible for maintenance of equilibrium and posture, and coordinating various movements and smoothing out the movements, so that the body movements are in a very coordinated and smooth fashion. The disruption would lead to disruption in the matching of the motor inputs from the motor plan that is sent to the cerebellum and the real-time proprioceptive inputs that it is receiving, and it is these that lead to the various signs and symptoms in terms of ataxia, intention tremor, past pointing is one such sign that we see, dysdiadochokinesia, and all those features. In the end, I'd like to say thank you for a patient listening, and hope that this lecture is useful to all of you. Thank you so much, and I would like to thank PSI once again for this. Thank you.
Good evening, everyone. So, we are at the end of the very fascinating lecture by Dr. Abhinav Dikshit on the physiology of cerebellum and the cerebellar diseases. Today, we have quite a good lot of questions put on. I think since this was a recorded session, uh, all the questions will surely be addressed by our uh, presenter today in the due course. Uh, and I'd like to first very much thank Professor Dikshit for this wonderful lecture, a very crisp lecture on both the physiological aspect as well as the clinical aspects. At the same time, I would like to thank the Physiological Society of India, who, along with the Physiologic Education Committee, has could continue with this excellent online lecture series, which would cater to the needs of both the UG and the PG students who are studying physiology, both the medical physiology as well as a basic degree in the human physiology. All the persons who were at the backstage, like Dr. Arno Banerjee, who is one of the members of the education committee, Dr. Kaushik Bharati, Dr. Moshumi Datta, and Dr. Dakshayani Mahapatro, all of with the constant effort of all of the members, along with our President Professor Proshan Priya Nayak, Vice President Dr. Kaushik Bharati, this online lecture platform wouldn't have been possible. I think if we have a good feedback from all the viewers about what kind of topics you want to listen from us, that would be that would enrich us to give more and more lectures to you. Thank you all. I hope you all enjoyed the session, and we come up with another topic in the coming month.
Thank you, Dr. Sampa. Just to before we conclude, I would like to mention that because the speaker is not available online live today, we will surely address these, send these questions to him, and we will like to give the reply, get the reply from him, and then and other members who are later will be watching this session later, they can contact him on his email ID. Thank you, everyone.